In Digestive System 2, we will continue our discussion of the alimentary tract, looking at the small intestine, large intestine, and we will also go into nutrition. Part 1, we will look at the small and large intestine structure and function. This is a review of what we looked in at the previous lecture. Small intestine is a long tubular organ all the way from the pyloric sphincter, that is the closing at the lower end of the stomach, to the beginning of the large intestine. It receives chyme from the stomach and it produces several digestive enzymes that helps digest the nutrients and absorb them into the body. There are three components: the duodenum, jejunum, and the ileum. We talked about what their structure and function were meant to do. And then we also discuss how the inner lining of the alimentary tract, the small intestine in particular, we had these interstitial villi finger-like projection that are meant to increase the surface area available so that more nutrients, more material can be absorbed. It also helps with the mixing action with those finger-like projection. Each of these villi consists of simple columnar epithelial, and they contain connective tissues that house blood vessels, lymph capillaries, and also they are called lactale, the lymph capillaries that are within this villi-like structures. Blood capillaries and the lactale transports the absorbed dietary nutrients out of the alimentary canal into the blood vessel system that will take them to the liver. Between bases of adjacent villi are tubular interstitial glands. So these glands are the ones that will excrete mucus, excrete the secretory enzymes that are being produced. Again, looking at the alimentary canal, the four layers that we talked about: mucosa, the innermost layer, epithelial cells forming these finger-like projections, the villi. Then we have the submucosa, the connective tissue layer with blood vessels, then the muscular layer mucosa, the serous layer, the outer connective tissue layer. Looking individually at the structure of an interstitial villi, these are these finger-like projection lined with columnar epithelial cells that also have certain secretory cells within them, and we see this lymph vessels and arteries and veins that go there so that they can both take oxygen and then bring the nutrients that are being absorbed. Looking at a histological section of these interstitial villi, again we see these finger-like projections, and then below them we see the submucosal layer, and then at the basis of this finger-like projection we have these interstitial glands. So these interstitial glands that are found at the base of these villi secrete large amount of watery fluid. They carry digestive products into the villi. So these digestive products are several different types. So basically they have enzymes that will help digest all different types of nutrients, proteins, sugars, and even lipids. So proteins are being broken down by peptidases. There are sucrases, maltases, and lactases. There are different types of sugars that are broken down disaccharides, meaning two groups of sugars broken down into monosaccharides, two individual sugar molecules. And then we have the lipases, interstitial lipases. So where it produces a lipase, it will have an associated name. And they again break down triglycerides into fatty acids and glycerol. If you remember from several lectures before, we studied that triglycerides are formed by 3 units of fatty acids with 1 unit of glycerol. So these interstitial secretions have to have a regulatory process. They are not being constantly put into the small intestine. So they are stimulated when there are mechanical and chemical breakdown of nutrients that are happening. So then there is mucus that is secreted to help with the traveling of the chyme or the food bolus. And when this alimentary canal extends or distends, that stimulates the parasympathetic refluxes, and that stimulates the secretion of the small intestine digestive enzymes. So again, remember the different digestive enzymes that are being secreted and produced in the different components. We started to see some in the mouth, then in the stomach, then also in by the pancreas that was put into the duodenum, and now we see there are secretions, digestive enzyme secretions produced even in the small intestine. So let's talk a little bit about the absorption in the small intestine. Small intestine is the major site of absorption within the alimentary canal. They absorb monosaccharides, that are individual sugars, amino acids, they are the components of proteins, and fatty acids, which are the components that come from lipids. So these then are collected into clusters. So these large clusters are called chylomicron. So those are, just think of them as lipids, almost lipid molecules, like a sphere of lipids that can just embed some of these nutrients and help transport them. So chylomicrons are the large molecules. Then you also have two other types known as VLDL and HDL. VLDL are very low density lipoprotein. So these are lipoprotein molecules that have a low density when it comes to the lipid content. So the VLDL also can have low density lipoproteins. They consist mainly of cholesterol. Then we also have the high density lipoproteins. These help remove cholesterol from the tissue. So when these lipid molecules are being absorbed from the digestive tract, there has to be a way for the cell to regulate, accumulating them, taking them somewhere, and also being able to remove them from the tissue. And we'll talk about these later on when we talk about the good types of cholesterol and the not so good types of cholesterol. Again, here looking at the process, we have these epithelial cells in the mucosa of the alimentary canal. Here are these are actually microvilli, very small finger-like projections that will be able to absorb the broken down nutrients that will go through these cells, get into these lipid molecules, chylomicrons, and these chylomicrons are then taken up by the lymph vessels, blood vessels, and transported them to the liver. So that's the purpose and function of the small intestine. So now let's discuss the large intestine. The large intestine, as the name says, large because it has a wide diameter. It's only about 1.5 meters long. It begins right after the end of the small intestine. It starts at the cecum, and we'll look at the structure, how this looks like, and it ends at the anus. So the main purpose of the large intestine is to absorb water and electrolytes and form and store feces. Large intestine composes of the cecum, colon, rectum, and anal canal. The cecum is the beginning portion. It is highly distended portion that has also this appendix projection that is a vestibule structure, not a proper function in the alimentary canal. Then we have the colon, which has several components, which we will look at next, which goes into the rectum and then into the anal canal, that is the opening to the outside. So even though we have mostly smooth muscles throughout the alimentary canal, once it gets to the anal canal, now we have voluntary muscles, internal and external sphincters that will control the passage of the feces that are collected. Here's a representation of the parts of the large intestine. It starts at the cecum, and then you have this ascending colon. This is the arrangement of the large intestine in our body. So you have an ascending colon, then you have a transverse colon, you have a descending colon that goes into a sigmoid colon, an S-shaped colon that goes to the uh, anal canal, followed into the anus. So here's the structures where we have the ileum that comes in and connects with the cecum right here. And then that's the structure that goes beyond in the large intestines. So the final two components after the sigmoid component are the rectum and the anal canal. So rectum is the pouch right before the anal canal, and then you have the anal canal that's supported by these external anal sphincters, which are skeletal muscles that we are able to control. So again, looking at the rectum, the anal canal, it still has the four different layers that we discussed throughout the alimentary canal. There are some modifications which we'll look at in the next slide. So what we see here is the rectal area, and then followed by these certain structures that are found. They are called the anal columns, and then we have the internal anal muscle, the sphincters that are on the inner component, and then we have the external anal sphincters that allow the passage of feces under our control. So looking at the structure again, we talked about how it has the four layers of the alimentary canal, but the unique characteristics it has is that large intestine does not have villi because it has no function in absorbing nutrients, therefore it doesn't have villi. It has three bands called the taeniae coli that extends the whole length of the colon. These longitudinal bands also help with the structure, maintaining the structure of the large intestine, and then these taeniae coli exert tension that creates a series of pouches called the haustra. And those are the ones that we saw right here. These particular pouches like thing, the haustra, are present in the large intestine. Function, we talked about how the large intestine does not absorb or digest any nutrients. It only produces mucus to try to help with the passage of the feces that are produced, but it does absorb electrolytes and water. It also houses a lot of important bacteria called the intestinal flora, and they help with the synthesis of certain vitamins, including vitamin K, vitamin B12, thiamine, riboflavin, and these vitamins are then absorbed and used in our body. So the final things I need to mention is feces. So the end result of the food that we ate, the broken down product that goes as the food bolus from the stomach, comes out as the chyme, then broken down further, the components that cannot be digested, cannot be absorbed, form the feces. So it contains undigested material, mostly water, electrolytes, mucus, any bacteria, sorry, any cells that were shed, that slopped off, any bacteria, and also feces has a certain pigment, the color that it has, from the bile pigments. Any bile that was not absorbed by the body, we talked about how it was produced in the liver, stored in the gallbladder, and used for the digestion of the food, gives its pigmentation, the color. And the odor is due to the chemical products produced by the bacteria in the colon area. We will pick it up from here and discuss nutrition, looking at the glucose glycogen cycle in the next part.